passion, intuition, discovery. Join Florida Planting as we celebrate the possibilities.
So congratulations to everyone who is involved in making that happen. It really is an outstanding achievement for our university. So moving on. In addition to research, we also are very proud to have established a neuroscience PhD program in 2021. And we also have several community programs where we partner with community organizations, and that's where my role comes in. One of these programs is called Brainy Days, which you are here for this evening. Brainy Days is a month-long event that we have in March where we celebrate neuroscience all month long and have featured speakers, including tonight's guest, Dr. Michael Burton. Our next speaker and our final speaker for 2025 is Dr. Andy Newberg. He will join us next week, Wednesday, March 26th in West Palm Beach at the Mandel Public Library. So if you can make it out there at 6 p.m., his talk will also be followed by a reception and a book signing. Please register if you can so we know how much food to get for everyone. There's a QR code there. You can grab something, a little postcard out in the hall as well. So now I would like to thank our sponsor, Palm Health Foundation, for their generous support of Brainy Days and introduce Pat McNamara, President and CEO of Palm Health Foundation.
Thanks so much, Nicole. So what I would like for you guys to do for just a minute is to imagine South Florida has become a brain-forward ecosystem and all that that entails. From the amazing neuroscience research that we have going on right here to engaging our community deeply in bi-directional communication, informing the research, and in turn, the researchers informing the community about what's going on, and fostering innovation. We see the potential of South Florida to become known globally as the Brain Coast. And so you'll be hearing a lot more about that. Thank you.
Thank you, Pat. Now I would like to introduce Dr. Ning Kwan, Professor of Biomedical Science in the Charles E. Schmidt College of Medicine.
I'd like to make this introduction to our invited speaker. We're very lucky to get Dr. Michael Burton to come over here. He's a little hard to get, actually. The first time we tried to get him, he suffered from a severe COVID infection. We couldn't get him. And finally, we got him for the Brainy Days, and I'm very happy that he's able to get this trip to give us this talk. And Dr. Burton is a rising star in our field of neuroimmunology, and he's currently the Eugene Derma Distinguished Professor in University of Texas in Dallas. I found out he actually had a lot of connection with me in terms of, well, I think I have to let you know how old I am, because his mentors, his PhD mentors, was like Ronald Johnson, a good old friend of mine, and then he did his postdoc. It was Joe Alquist, another old buddy of mine. But now he has his own research. In a short period of time, he has published quite a few papers. Like in the last 5 years, over time, he's close to 50 publications, and highly productive. And he's also pioneered some of the areas of the neuroimmunology connecting the immune system, sensory system, and the pain, concentration and pain. And over this short period of time, we've gotten a lot of awards from NIH and NSF. And he was also awarded for outstanding mentorship and outstanding research achievements. And he even got a congressional recognition, certificate of congressional recognition. And he's a very, very good speaker. We have a science talk this afternoon, and I'm sure we will enjoy this talk for this hour. Mike.
Now that that happened, good afternoon, everybody. Afternoon still. First off, thank you so much, Ning, for the invitation to come and give a talk. Actually, two talks today, so I feel super special because I get to double talk. So I always like to kind of open up these types of conversations and kind of let people know a little bit about myself, why I'm here, why I study what I study, and what brings me kind of excitement. I think putting perspective is always important. So I talk a little bit about my background. I'm from a single parent household in Chicago where we thought the only way to get out of the inner city was to play sports. And so I did that in high school, and I ended up getting to undergraduate and walking onto the football team at the University of Illinois at Urbana-Champaign. I learned really quickly that it was really hard to do, but I was able to earn myself a scholarship after about a year. And trying to keep that scholarship, I actually got a really, really terrible injury where I herniated a disc in my cervical vertebrae, which essentially meant that I could no longer play. I had severe inflammation and severe pain that lasted for quite a while. And once the inflammation cleared up, I still had pain that lasted about 6 months to a year afterwards. And it left me really wondering, why me? Why this? Another thing that was really important, which was really sad, is as fast as I earned that scholarship, the scholarship dried up. I didn't even get to cash in on my tuition waiver for the semester. That's how fast I got hurt. And so I realized I needed to really rely on myself. And in doing that, I decided to get into a lab and try to understand what is going on with my body, why did I have this inflammation, now that this inflammation is now gone, why am I still in pain? So I ended up working with Rodney Johnson, I guess and met Ning, my scientific uncle, if you will. And we kind of delved into understanding how these cells of the immune system interact with the cells of the brain to elicit various behaviors, in particular, pain and cognition. I ended up moving down to Dallas to start something called my postdoc, and I worked with two really huge people in the field of pain and inflammation and neuroanatomy. And there's a saying, I grew up in Chicago, but I got to Dallas as soon as I possibly could. The students love hearing me say that because they want to hear people that are from Texas and from Dallas. And I've been there for about a decade now, and I've been having a blast doing what I do and having my own lab space. Okay, so the lab space. I opened up the lab in 2017, and this big, beautiful building where you can see all the windows in the back, if you can see. However, my lab's unfortunately in the basement. I don't get to see these fun windows, but we get to go to the top floor. Levels of trainees, so we have undergraduates, high schoolers even that come and get involved into the lab, medical students, postdocs, all types of individuals who engage in not just training the next individuals for science, but just next individuals in the community as well. So we have a lab philosophy I also like to share with people, and it's don't be evil. And the reason why it's don't be evil is based off of the early days of Google. And I'm not sure if people remember this, but their first slogan was, you have all the information and access to it in the world, but just don't use it for evil. And we kind of see science the same way. I want everybody to have as much information as possible and resources as possible, but just don't use it for evil, use it for good. I ask that my individuals in the lab be as deliberate as possible, not to be indifferent. Indifference to evil is worse than the actual evil itself. In a society of free individuals where there's choice, while there may be some people that are guilty of certain things, all are responsible. And I think it's a really interesting message to send, you know? And also good science is for everyone and it should be shared aptly. And so that's kind of how I feel about what we do and what the science we do. Now, we work in a preclinical to translational setting, and what that essentially means is we work with preclinical models, basic models, looking at how the systems and cells in the systems are really important to mediate responses. But then we also try to translate that to actually say, what does it mean for you? What does it mean for everybody else? And to do that, we take this integrative approach. And we want to understand how lifestyle factors like age, sex, diet, and alcohol, which we'll talk about today, influence the communication between these complex systems of the nervous system and the immune system. So my lab study is something called psychoneuroimmunology. It's a mouthful. We shorten it to neuroimmunology, and it's really taking the fields of immunology, psychology, and neuroscience, and putting them up in a blender and putting myself right in the middle of it. And the studies are really what happens when the immune system impacts the nervous system. The immune system gets activated. Usually the best example I have is when someone has a cold. When you have a cold, you change your behavior. You decide that you want to sit in the house, you want to rest up, you don't want to eat as much. Your immune system is telling your brain, I need to rest up and do these things to get better. And so the study of neuroimmunology is having this immunostat, essentially, where I want to make sure my body and my brain and my behavior all behave nicely. So the focus of our lab is where we really, really are interested in pain. And so there is two different kinds of pain. One is normal. I need to know that I'm in danger. My hand is on a burning stove. This is too cold to hold. That rhymed. But then the other kind of pain, which is something that I experienced, is I had an injury, and the injury lasted for a little bit of time, but then the pain actually resulted, or the inflammation resolved, and it resulted in even more pain later. And that's chronic or maladaptive pain. And interestingly about that, there's kind of two parts to it. One part is the pain experience itself, meaning what is actually this thing causing my pain, but then the processing that's involved. And there's a number of factors, from psychological factors, for instance, being a tough guy, being an athlete. I can't be in pain, even though I look down and I see my bone coming out of my skin, right? It's something that I can't think about, but in fact, I am in pain. There's other factors like my genetics, comorbidities, like if I'm already depressed. When I'm depressed, I can feel, and when we are depressed as individuals, we do feel more pain, and it's hard to tease out between depression and pain. And then there's also social factors as well. Where do I fit in society? Am I a man? Am I a woman? And so the focus of the lab is trying to take all those factors on top of the normal input that you have with pain and put it all together and try to understand how we can figure out what's going on. So kind of a brief outline today is we'll talk about a few things. One, the problem, which is pain. I'll ruin the punchline. Some of the approaches that we use in the lab to study this, and then also looking at alcohol-induced pain states. It's a tough topic to talk about because everybody loves a little bit of alcohol. And then we'll talk about cells interactions that are included in alcohol-induced behaviors. And so the major problem, super simple, 50 million Americans suffer from chronic pain. Interestingly enough, 70% of these people are women or female. 25% of pain patients that are prescribed opioids misuse them, meaning they abuse them. They don't work so much anymore, so they take more of them, and then they overdose on them. And this ends up in opioid-involved deaths. And in about 2021, there was roughly 121,000 opioid-induced deaths in the country. And so we would say we were in the midst of something called the opioid epidemic. So the thoughts are with this problem is, can we discover new ways to treat pain that are non-opioid based? So our approach is really, we care about all the pain pathways, right? I want to know why that stove is hot. I want to know why that pole is cold. But I also want to understand, how do I get from this cold, hot, normal state to this chronic, bad or maladaptive state? And to do this, we want to understand the unique conversation that happens in between the nervous system and the immune system, because it's a clear conversation. Pat, you said bidirectional. There's definitely a bidirectional communication between immune cells and non-neuronal cells. And then we want to identify new drugs that can target cells that can alleviate these pain states. And then we want to understand essentially why pain influences women more than men, right? 70% of individuals that experience chronic pain are females. And so delving into this alcohol world, and so I am oftentimes conflicted because I was doing a podcast earlier and I was asked what's one of my favorite things to do, and I said drink whiskey. But then we started doing this experiment looking at the context of what happens when you drink alcohol and how it changes your body, how it changes your physiology. But we take a step back and just look at alcohol and inflammation across the world. What we found is that individuals from over 150 countries, there is an upregulation or way more people drinking alcohol, but then having alcohol-related inflammation, alcohol-related comorbidities like depression. And if you look at some of the higher numbers, you see America, China, India, Russia, Brazil are in the top kind of countries with that higher amount of prevalence. And that prevalence is 27% increase across time. Over the past 30 years, again, over 150 countries have a 27% increase. Interestingly enough, more people around the world have comorbidities with pain and alcohol, which is depression. And approximately 40 million people, if you look at just individuals that drink alcohol, are individuals that deal with chronic pain and alcohol. So clearly there are multiple factors that contribute to pain states, alcohol consumption, injuries, et cetera. And so we really are interested in trying to understand and tease apart what's going on. So we are familiar with everything is good in moderation spiel. A little bit of something is good, too much of it is bad. And have a few, I guess, articles from the New York Times over the past just 4 years. And the first one I have up here is one from 2023 that talks about the battle over what to tell Americans about drinking. There's some health hazards in amounts, in how much or how little, what's good, what's not. And this one in 2023 came out, even a little alcohol can harm your health. And so, interestingly enough, there's been decades and decades of confusing or back and forth communication with what's good and what's not. Things are good in moderation. A little bit of alcohol is good in moderation. Too much is bad. But in fact, it seems as if even a little bit can be deleterious. And as of earlier this year, the Surgeon General actually calls to put a warning on alcohol bottles that alcohol can cause cancer. Who's heard about that out of curiosity? Okay, a few people. And so clearly there is some long-term research that has been done and some long-term research that still needs to be done to really try to understand what's going on. But if the Surgeon General is calling for this alcohol warning, there's something that we have to really heed to. And so the reason why that's important is the human condition. And when I say human condition, when we think about the research that we do preclinically, we put together using animal models or other models to try to control everything. However, the human condition is you can't really control it. You have someone that has a diet that eats this, they drink that, they're stressed, they're inflamed for different reasons. So it's really, really complicated and really, really difficult to try to use human subjects in a lot of cases. And the biggest thing is there's something called attrition, which is we can recruit people to studies, but then they just leave the study or they drop out of the study because it just doesn't fit them for whatever reason. And so it's really, really hard. And this slide up here is talking about time and how timing is really, really important. It's important because when you think about a pathology, by the time you get to an actual diagnosis and it's straightforward, you're really far gone, meaning you're, severe Alzheimer's, severe Parkinson's, and it's really difficult to reverse these things. And so by the time we get to that pathology, we're kind of late. So what we want to do is try to look at other unspecified overlapping symptoms like pain, for instance, and try to understand if there a way to tell, oh, that tweak that I felt or the fact that I'm having more pain to this particular stimulus when I shouldn't for a reason, right? Is there something I should be looking a little bit deeper into? Because the idea for pain is it's supposed to be a notification, hey, there's something going on. There's something a little bit off. And so what we want to do is use these overlapping systems and try to understand, like for instance, in the context of chronic pain, it's been shown that in Parkinson's and other neurodegenerative diseases, that chronic pain is an early detection marker or early symptom for understanding that there is a development process happening, but it gets ignored because it gets wrapped up into many, many other unspecified conditions. So we take the approach of the road less traveled, and it's been really a struggle because usually researchers go and say, I want to look at developed pain states, develop chronic pain states, and try to reverse it. But again, most of the time, it's way too hard to try to do. So can we look at it earlier, before the onset of chronic pain, to determine if there's mechanisms behind its development? So can I look here and not necessarily talk about chronic pain, but what if I just have changes to sensitivity? Like my pain is not necessarily in a real bad way, but I do realize, hey man, I usually don't feel this way when I wake up in the morning. Oh, hey, I just stubbed my toe and it's still hurting this morning, and it's this afternoon and I still don't feel well. These types of things is what we're trying to do and trying to uncover. And so this is kind of an interesting slide. I like it, but it kind of talks about kind of the alcohol habits that we have, and then some of the different pain conditions that we deal with. And it's kind of a two-parter here. On this left-hand part, it's really, really clear that surgery, low back pain, fibromyalgia, there's tons and tons of things that cause pain. However, on the top here, we think about drinking short-term or short and small amounts of alcohol. What we'd like to do and try to understand is not necessarily pathological alcoholism, but just a couple of drinks. Maybe we call it the spring break model, right? Where for a short period of time, I'm having some drinks, but I'm not necessarily a pathological drinker. Or another one we call is the soccer mom or the wine mom culture. Does short-term moderate alcohol consumption alter pain sensitivity or elicit something we call allodynia? It's just a medical term for changes in pain sensitivity. I feel pain when I shouldn't feel pain anymore, or I shouldn't feel pain at all. And so we have this model where alcohol drinking can push our system from acute to chronic pain. And so in our model, we have this kind of liquid acclimation of giving an animal alcohol. And what we find is that this low dose alcohol consumption leads to something called an increase in priming and no pain. However, you stub your toe or something that's a super low threshold that shouldn't cause a ton of pain, you see a gigantic response that lasts, but then kind of resolved itself. And independent, or I guess without having this alcohol, you don't see this actual priming. And so in order to kind of look at that, we had an objective. And the objective was, does this short-term pain or some short-term alcohol moderate alcohol-induced pain sensitivity? And then are there changes in allodynia, meaning are there changes or lowering in our pain thresholds? And so in order to do that, we looked at something called hyperalgesic priming. And it's kind of a mouthful, but essentially what it means is if I take a normal paw or normal skin, and then I inject a low amount of a subthreshold dose of something that causes inflammation, I get something acute because you just injected me, but I'm okay. However, if I've been drinking or I've had a bad diet or I'm just in a prime state at some point, but then I inject that same subthreshold dose that only causes a minute response, it's actually an exaggerated or way greater response, and it lasts for way longer. And why that's important, again, is trying to understand that transition from this acute to long-term chronic transitionary pain states. And so this cute little mouse model we use is called a liquid diet ethanol model or alcohol consumption model. We have a nutritionally complete diet. The only thing is that we add a small percentage of ethanol, which is about 5% ethanol, and we let them drink the ethanol for a course of about 20 days. That 5% ethanol diet is similar to drinking about 1 beer a day or a glass of wine a day. And then we give the animals their time to hang out, and then we do diet placement, and we do records, and then we look at pain behavior. And I'll show and talk a little bit about some of the pain behavior that we look at, and then some of the affective behavior, which is comorbidities or depression-like behaviors. And the student that did some of this work in the lab is Siobhan Alexander. She just received her PhD and is doing her postdoc at Tulane right now. Super proud of her. But so just to remind you all, the idea is that during this liquid diet acclimatization phase, you are letting the animals get used to it and then prime, and then you give them this kind of subthreshold dose, and you see this effect. And so here goes an example of where we use just normal wild type animals, and we wanted to understand males on the left, females on the right. What's the effect size of giving this ethanol and looking to see if it changed pain sensitivity? As you go up, you get more pain sensitivity, and as far down as you are, you get less. Interestingly and scarily, what we found is that with this kind of short-term ethanol regimen, that females experience way more pain on ethanol than their male counterparts. And we call this mechanical sensitivity. A mouse can't tell you I'm a 1 out of 10 or 5 out of 10 in the context of a pain scale. So we have to do something called mechanical sensation. And what you do is you just take a filament, which looks like a toothbrush filament that weighs about 1 gram, and you apply pressure on the bottom of their paw. When they feel pain, they move their paw a lot quicker than they would if they don't. And if you look at the effect size, you see this is a greater kind of output for that. And so the question that we had about that was, are the neurons affected, or the cells that sense pain are affected, or is it the immune cells that are affected, which are the cells that are around the neurons? And we had reason to believe that it wasn't the neurons necessarily, but the non-neuronal cells. And so we looked at a place called the spinal cord, and we looked for these immune cells, and we just counted them. More immune cells means more inflammation. More inflammation means more pain. And we found that in the females, there was a consistent and significant more amount of these immune cells that directly related to the amount of pain that we saw. So drinking this ethanol diet didn't just change how you felt pain, but it actually activated your immune system just the same in a female-specific fashion. And so these are a lot of words, but we wanted to understand what about these immune cells in the spinal cord that were really important for mediating this. And we did a literature search, and we found that there's a particular gene that binds directly to the alcohol called toll-like receptor 4. Why does it bind directly to the actual alcohol? There is something called the motif, or what's the best word I can use? It's the alcohol kind of binds like a lock and key mechanism and drive or activates the toll-like receptor. It's expressed on immune cells. It's expressed on neurons as well. And so we wanted to understand, looking at the previous literature, if we have alcohol and we give these animals alcohol, these cells are called microglia, what happens in the context of activating the microglia and leading to neurodegeneration in ethanol or alcohol-induced behaviors, anxiety, and addiction. So the idea, again, is what are these immune cells that are found in the spinal cord and also the brain? When you activate them, how are you getting these downstream effects of alcohol, addiction, anxiety, pain, et cetera? And we had a hypothesis is that alcohol and also its metabolites are important to activate these immune cells, microglia, and drive the initiation of alcohol-induced pain in females and females only. The problem with this particular receptor is it binds to the alcohol, but it's on a ton of different cells. And one of the things that I said earlier we really want to do is try to identify a cell that we can target so that we can treat the actual cell to prevent pain. But this particular receptor is kind of on all kinds of cell. It's really, really hard to target. And so we really want to understand a way to make it so that this cell and this receptor really matter. And so in order to do that, we use something called a requirement versus efficiency model. And the long game here is remove that particular receptor from all the other cells. So remove the noise. I don't want to know what is doing in any other cell but this particular immune cell that's supposed to be important. And why that's important is because I want to understand, can I target this cell to really help our animals and our people and long-term in the context of preventing alcohol-induced pain states. So this is a lot, but at the end of the day, what I wanted to really show is that we used this method to truly target these cells that are in the spinal cord and say, hey, I want to take this particular gene, I want to remove it, I want to put it only in our cell type of interest. And we were able to do that. And as scientists, we got to prove like 16 different things at one time. And so I'm just showing you kind of all of it at once. So the first thing we wanted to do was make sure that the cells of interest express the receptor of interest. And the receptor of interest is the TLR4. And long story short is what we had our normal animals, and we compared them to our genetically altered animals that only had the receptor in our cell of interest. They look the same. And the reason why we wanted them to look the same is we didn't want any differences in the context of how that receptor recognizes the alcohol. I want to put this receptor on one cell type of interest, and I want to activate that cell with alcohol, and I want to see, do I really see a painful state? So we did this experiment with these animals, and I'm going to show you a different looking graph this go around. And this graph is looking at the same kind of withdrawal threshold, but it's a little bit reversed. More pain, the lower you go. So the lower the actual gram threshold, that means the animals are responding way quicker. The higher the gram threshold, the animals are not responding whatsoever. And what we find is that in our animals that have the receptor only in our immune cells, that the females are actually the exact same as their wild type counterparts. And so at the end of the day, when we remove that particular receptor, they didn't do anything. So we know for a fact that that receptor is targetable. But then when we put that gene back in that cell, it's actually showing similarities between the wild type and the reactivated animal, or the animal that had the receptor only in that particular immune cell. And why that's important is now we can show and prove that these cells are in fact targetable, and we actually know the exact gene that's targetable to help individuals in the context of alcohol-induced inflammation. And this is just to show that when you have male data, so the previous data was females, but this is the males, and for some reason they're flatline, and then when you compare to a female, the females are all the way down here, and that's to show that there's a clear difference between male versus female animals. And we did the same thing where we looked at these activated immune cells in the spinal cord, and what we found was that there was way more active immune cells in our female groups compared to our male groups. If we look at our y-axes, we have up to 50 to 60 versus 30 and below 20 in the males over here. And so clearly the ethanol is getting into, or the alcohol is getting into the immune system, activating these cells, but it's only doing it in a female-specific fashion. And so at the end of it all, we summarize it with there's female-specific pain sensitivity, there's more immune cells that are active, and that we looked and we saw that there is a particular cell in the spinal cord that's really important for this activity. The next thing we wanted to ask is, what happens in the context of this pain when you think about depression? Like I was talking about earlier, there's a 27% increase across alcohol drinking and prevalence of alcohol pathologies in 150 countries across the past 30 years. And that prevalence is depression. And we're curious, does activating these same immune cells that cause this pain also cause depression or depressive-like behaviors? And in order to do that in mice, again, we can't ask the mouse, hey, are you depressed? If I could, this would be really easy. Be really, really easy. But instead, we have to do a couple of different assays to kind of tease out what's going on. So we put the animals in the same kind of paradigm where we let them have ethanol for about 14 days. And then we ask them, how much or how immobile are you? And the immobility factor is what we call learned helplessness. When you're depressed, there's about 5 different measures or modifiers that you have to have. One is you feel depressed. Another one is you are don't have as much energy. Another one is you just don't want to get out of bed, right? And that's kind of the equivalent to learned helplessness in a mouse. We call it immobility because they just don't want to move, even though they have all the motivation to move. And when we look at our animals at the 20 days after their ethanol or their alcohol paradigm, what we find is that there's not a big difference. There's a bunch of different things happening, but there's really no difference across the board with the males. But in the females, we see that at day 20, there is an increase in I don't want to get out of bed syndrome, right? There's an increase in immobility in the females. So there's kind of this kind of cross talk between the development of pain and also depression. So some concluding thoughts to all this is that putting your animals or yourself on this ethanol diet can cause this increase in priming across time, and being exposed to something that's super low threshold or sub-threshold can lead to an inflamed or other pain state that you would normally not have. And this kind of says there's a clear neuroimmune conversation between non-neuronal cells or immune cells and then neurons. This particular receptor on microglia do drive pain behavior and depression. People always ask me, what about other cells? Since you say there's so many other cells in the mix, they're undoubtedly involved, but I need more money and time in a day to look at all of them to be able to do that. And so, but we do feel really excited about the idea that we found a potential targetable cell type and a targetable pathway to help with understanding not just pain, but depressive-like behaviors as well. And so with that, I like to show kind of the lab and the progression of the lab over the years. There's just a fun group of people to work with. Dare I say a diverse group of people as well. I think it's been a fantastic journey. I've been around for about 7 years. The lab's been open for about 7 years, and I've been able to graduate 7 PhD students and have several postdocs, people after their PhD, come and work in the lab as well. Just a great, great place to be. And I have a cursory where if people want to follow me on Twitter, if you're still on Twitter or X, it's down here. And I'm also on Blue Sky. I added that. And here goes some just people that I work with across the board, which have been some fantastic collaborators. We have funding from the NIH, from a couple different institutes, the National Institutes of General Medical Sciences, and the National Institutes of Diabetes and Digestive and Kidney Disease, a couple other small funding sources like the Mayday Fund, the Rita Allen Foundation, and some industry funding from a company called Abbott. And thank you guys for your attention. Appreciate it.
Thank you, Michael. So we've got a couple of microphones here, and we did ask Michael to save time for discussion. And you've opened up a bee's nest of questions. So we're going to start there. I'll start with one, and then we'll start going around the room. So with the female effects that you saw, and some were quite strong, do you look at the development over time? Is it, do you have to have, is it just that one drink once, or is it that one drink 100 times? And then, do you see a time dependence to this acquisition of this heightened pain state?
Yeah, that's actually a great question, Randy. So, it's kind kind of deep too. Always the waters run deep. So if we look at across time, day by day, there is no development until about week 2 or day 14 in terms of this alcohol-induced change. Interestingly, if we keep going and we don't do anything to the animals, about 4 weeks later, they actually start to develop small amounts of pain independent of that subthreshold dose. And other people have also published that as well, that if you drink, and that's equivalent to 4 weeks in a mouse is equivalent to quite a few years of time in a human. And so but that's every day. So if that answers the question.
Yeah, I think so. I think, this time dependence and, abstinence and that, maybe I'll go to that.
Yeah.
How about
yeah.
abstinence?
Yeah,
Have you-
good. Another great question. And so, can I help myself? Can I throw out this drink today, put this drink down and be fine? Yeah. So we've tried, and that priming effect where we see the changes in the pain behavior last for about 6 weeks, which pretty significant amount of time. So it doesn't mean that it's not reversible. It means it just could take more time to reverse.
Thank you very much. Have you or your colleagues found any connection or studied what the hormonal milieu does? I mean, is it the same response in postmenopausal women and premenopausal women, or hypotestosteronism in men? Do they respond the same way?
Yeah, good question. I love the question. So a couple of things. We looked at the cycle and hormonal changes across those 14 to 19 days, and we saw no difference with that in this model. Also, in human, that's also been shown as well that there is zero, next to no correlation with fluctuation in hormones in the context of this type of behavior, which is crazy to think about, right? Because the first thing you think about, what's the difference between males and females? However, I do have an answer for you, because I have to have an answer, because it's a question I get every time I go out places. And the answer is, so especially in humans, you have development of your nervous system, and it happens when you're still in the womb. And there is essentially a way that your body gets programmed one way or another way in the context of the hormones that are involved in development. And we think that it's a programming problem, not necessarily once you're done programming, if that makes any sense, right? So if you are in the womb and born female, essentially we think that you're going to be susceptible to this kind of reaction no matter what. And it just has to do with hardwiring, not necessarily the changes that kind of fluctuate throughout the month from month to month, which is why you can't find it, which is why it's not super duper obvious, right? Because if it was really hormonal changes across the month, I think women would know way more, right? And I think that's a logical answer. I don't know if that's true, but that's the answer I have. So, yeah, no worries.
Curious if any research has been done when there is known pain in the subjects, right? And you start with the pain already there, and then you look at the response and back away from that by adding the alcohol or not adding the alcohol.
Yeah, that's a fantastic question. And I think that's part of the problem of why this is going to be a hard thing to study. And there's a two-parter, and the first part is if you were to look at the animals and you just looked at them in a cage and you poke them, independent of that subthreshold dose and pushing the system, they don't really feel any different. They're not in any pain whatsoever. So I don't have any reason to want to study. I don't have any reason to want to bring you into the clinic, right? People want to go to seek medical attention because they have something called spontaneous pain, which is essentially, this is hurting. Why is this hurting? And I'm always thinking about it, right? That's going to get you to the clinic. That's going to get you to seek medical attention. Otherwise, it's going to be really hard to do. And it's something that we've been battling with. Oh, she has more. Yeah.
When you've had a serious back injury, pain lingers.
Yeah.
That disc pain is real. And I know many other people that have had that. And so I was curious if the mice themselves Sounds cruel, but that pain could be induced in them so that it's consistent in an A and a B test.
Got you. Yeah. So we are interested in that down the line. But yeah, that's a good question. Can the alcohol exacerbate already existing pain? Which I was going to get to that point. We believe more than likely, yes, but it's hard to tease out. But it's a little cruel. We don't want to do that necessarily. But I will say, I have another answer for you. So the human condition is more than just I drink this one glass of wine a day. It's probably also I have a horrible diet as well in some circumstances. And so we do have a study where we look at individuals that don't have the best diet, have a horrible diet, essentially the McDonald's diet, and individuals that don't. And we see some differences in the context of pain being induced when you have a bad diet and you're drinking too much as well. And some preliminary stuff with that. So I would have reason to believe that you would exacerbate the pain state with the ethanol and that model, but we just haven't done it. So
Perfect segue. I was going to ask about, we naturally produce ethanol with the gut biome. Does that have an additive effect, or do you see these effects without, administering alcohol just as the body naturally produces it?
ooh, you know some stuff about the biome. That's cool. So this is a great question that I've never gotten. The first thought that comes to mind is I'm not sure how much ethanol or byproducts is actually produced from the microbiome, but I'm going to get that answer and figure that out and see how equitable it is to having a drink a day, right? Because you're right, the byproduct of bugs in our gut is going to have acetaldehyde or other metabolites that are similar to ethanol. Yeah, that's another great point. How I'll answer that one is thinking about the individual populations that eat those certain objects or eat those certain foodstuffs don't seem to have different amounts of pain or different or changes in their pain sensitivity, because there have been worldwide studies and people looking at pain sensitivity. I would say pain sensitivity. I would say that, or threshold to pain thresholds. So, good question. I want to look that up and I want to know that answer to that. And really, it's how much ethanol is in the blood in general. And so, what I will say, I'll give you another little tidbit of information that you may not want to know. But we looked at if the ethanol itself caused leaky gut and leaky blood-brain barrier in our animals, because we were curious, is the ethanol just leaking across, or the alcohol leaking across the blood-brain barrier, or is it causing a shift where you have bugs from the gut getting into the blood, and that's what's causing more inflammation and pain? And we didn't see, in this model, we didn't see any of that. So that kind of answers it, not at all, but some information that maybe you think is interesting. Go ahead, Randy.
I'm showing my age, but I was in the field of gerontology studying aging and Alzheimer's when they started advocating for a dose of alcohol a day, particularly red wine. Do you know if those early reviews even looked at females, given that they were 70s and 80s, probably had no females?
Good question. I have an answer to it, but not that exact answer. Again, sorry, but I would imagine not as much. I think some of the studies, especially pre-2000, didn't really have a focus with rounding up females, half the population. So we could have a good guess. But what I will say, though, is most of those countries that say, or most of those studies that say you should drink a glass of red wine, are countries that make red wine. Right. So like we went back and we looked, and it was like France and Italian groups and Turkish groups. And it was like, oh, who's actually looking at this in countries that don't do this? Not a ton. So I think that's helpful. So.
Got a question here.
Yeah. Oh. Hmm. Hmm. Hmm.
Okay, got a question back here.
One question. So, do you think that if you increase the alcohol concentration, would you see the same effect that you see in the female in the males?
Males. Yeah, that's a great question. I get that all the time, right? It's because it's not just all is lost and only females are sensitive. Is it that we're titering down or we don't give enough to both sexes to actually see what's going on? And so we actually have an active grant application where we want to find that answer, because it's kind of to Randy's question about timeline. Is there a time when we can give it where it becomes just painful regardless, or is there a time when it turns on where we can look at individual changes in plasticity? Also, are there times when you could be abstinent and recover? And also, can you change the model altogether and it be a binge model? What happens if you don't drink across days, but you just drink on the weekends? Weekend warriors, right? And so we're really interested in trying to get into all of that. So, but that's a great question. And my answer, my thoughts would be yes, you would definitely get to a point where both sexes are sensitive with the amount of alcohol that you're giving.
And maybe a trivial question, but related. In your males and female mice, do they metabolize ethanol equivalently?
Good question. Great question. So I also have a piece of data. Actually, if it's a lot, but we did all that. And so I'm going to just show it because he asked, not because I wanted to do the humble flex. But we saw that the consumption of alcohol was the same across the board, males over here, females over here. And your question is the amount of alcohol in the blood. And we found that independent of genotype, and this is all ethanol groups, they had all the same amount of ethanol across the board. So,
okay.
Yeah.
Well, now you got me going. So I got another. I have a better question and again a nerdy one. So, you showed us that if you eliminated a particular receptor on a particular cell type, you lost this pain sensitivity in the females, but you didn't. I think you just didn't have time. I think the data was there about the depressive behavior.
Yeah.
So was the same experiment capable of eliminating that too?
Yeah, great. You're right. It wasn't clear. So yeah, we were taking the same animals and asking, are you in pain? And then asking, are you depressed? Mm-hmm. And there's a stark correlation, like almost one-to-one correlation with the more pain you're experiencing or the animals were experiencing, the higher they were on that immobility or I don't want to get out of bed scale.
Mm-hmm. Okay.
So, yeah.
And then on your very first slide, your title slide, so you would pay attention. On your title slide, looks like you have an image of neuronal activation there, right?
Yeah.
You might want to go back because it's very pretty for people to see it.
Yeah. It was actually kind of it was kind of it was kind of I didn't talk about any of the data.
Right. So those little dots, yeah.
Yeah.
So those are activated neurons, right?
Yeah. So we did a section through the brain of the same animals that had depression, and we found that these particular areas, hippocampus, prefrontal cortex, and some thalamic regions as well, were activated.
So do you see an overlap then with the ones that develop this pain state with the one with the depressive behavior? Is that those go?
Yeah. We only looked at the animals that had the one-to-one or a really high correlation with depression. But then when we counted all of them out, let's say there's 20, 15 of them are one-to-one, and the other 5 are just some gradation in the middle.
Some noise.
It's pretty clean actually. So.
Here we go.
So many pain syndromes, especially something like fibromyalgia, clinically is treated with SSRIs or selective serotonin reuptake inhibitors. Mm-hmm. Is there any research showing that serotonin has any role in modulation of the toll-like receptor or expression of the toll-like receptor?
Ooh, that's a phenomenal question that I actually have zero answer for, but I'm definitely going to look that one up.
That one's in my wheelhouse.
I was going to say, would you know that answer?
So I'm a serotonin researcher.
Yeah.
And we found that the serotonin neurons are densely populated with these immune receptors that he's talking about, as well as another one that responds to a slightly different molecule, but is very important in your immune system as well. So there may be something more than just sort of a psychic effect. It may come down to where those molecules are located. And serotonin neurons are chock full of them.
Yeah. Yeah. I wish I had all the answers, but Randy does.
We wouldn't have anything to do, right? Got one there.
Great presentation, by the way.
Oh, thank you.
I was wondering if long-term low-dose alcohol use can be a trigger for autoimmune disorders similar to viruses like Epstein-Barr or commonly known as mono. And I also wanted to know if by any chance you've looked into whether we see the same priming effects on people with autobrewery syndrome or intersex populations.
Ooh, that's a mouthful. So the first question is, does long-term low-dose ethanol look like low-dose inflammation, right? Like a virus? We think so, actually. We hypothesize that after a certain amount of time, I was showing how there's no differences in the leakiness of the blood-brain barrier. I think that's going to, in the gut barrier, we think that's going to change as time progresses, right? And then when that happens, you're going to 100% get inflammation from gut bugs that will lead to long-term inflammation, long-term inflammation and long-term pain states. What was the other question? I'm so sorry. No clue. I got nothing for that one either. You guys are stumping me. Usually I come correct. It was about intersex, and so you want to.
Oh, yes, yes. So my question was if we see the same priming effects on people with autobrewery syndrome, which is where you produce ethanol in the gut microbiome, and then on intersex populations where you might see differences because their programming in development is different.
Right, and I don't really have an answer for you. It kind of based off of the thoughts that we're talking about, where the programming is in development is really what's driving what we think are the differences, the sex differences. And so if you take that out or you like switch and give different hormones to the different sex or try to do things like that, there's actually an experiment that we propose to do and our recently submitted NIH grant that you should tell your congressman to please don't do anything crazy with the NIH and funds. But because we think it'd be really interesting. But like I said before, the fact that we've looked at fluctuations in the hormones across estrous cycle, and we looked at not just the cycle, but like the actual hormones themselves, estrogen, progesterone, we don't see any real differences. So we don't think so, but we don't know until we try.
Thank you.
Yeah.
Well, Michael, you've done yeoman's work today with giving now your second lecture. This was a lot of fun, and thank you guys for coming to Brainy Days again. And remember, we have one next week. And for those of you who have picked one up, we have an annual magazine called Masterminds that give you a little bit more of a sense of what the Brain Institute does and its students. And we thank you for coming. So we have some reception outside.
Yeah, those are great questions. Yeah, they were fantastic questions. Those are phenomenal questions. Yeah, they're great. So that's a fantastic question. We're underpowered, meaning we don't have enough subjects to get to that answer yet. When we do the analyses to try to figure out how many animals we need quadruple the amount of animals. And so to me, that means probably not as much as you think or that you would expect, but I think it's because animals are so like clear and clean, so we can do it in a human population 100%. These are very inbreds, but they're very, very similar. Yeah, yeah. Thank you for your question. I appreciate it. Thank you. Okay. Mm. Mm. Yeah. Yeah. So first off, I understand more so. I was just telling Ning this that my wife's pregnant with our first kid, and I'm seeing the changes happening in front of my eyes, right? And so, and before then, I hadn't thought about it much, but thinking about it, I think it's a really great question. I would imagine that there's something 100% different about that. However, right? That population doesn't exist, right? Because if you're pregnant, you don't drink alcohol, right? Oh, well, you shouldn't, right? Yeah, yeah, but yeah. Opposite, yeah, no, I agree. You're right about that. I think evolution and evolutionary roles like um, caretaking or hunter gathering, we actually think those are important. Like for instance, in the hunter gathering aspect, if you are male, we think that you have to stay active on the hunt. So if you injure yourself, you can't be in pain. You got to get back to the village, and so there's a delay in male responses, but still, even though we're not hunting and gathering anymore, but it's still happening now because we still haven't evolved past that point, right? And what we're doing is actually really fun. We're looking at different species that where the hunter-gatherer roles are reverse and try to understand if there's more or less pain in the males and females that way. So we're doing it in lions. We have two lion dorsal ganglia or spinal cords that we're looking at and trying to understand if there's a difference or flipping in how those things are expressed. Getting lion tissue is impossible, right? So we got the Nebraska Zoo gave us a small piece. Yeah, yeah, small piece. I thank you for that one. Yeah. Really? Yeah. 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 Thank you. I appreciate that. Thank you. So nice meeting you, Michael. Are you with them? Okay. Fair enough. Hello. Thank you. Okay. Oh, okay. Got you. Got you. Yeah. I'm like, what's your question? What are you doing? I love your hat. Yeah, no, I find all these yeah, yeah.
the intersection. I had a similar question for you. I couldn't figure out that question. Like, but how can we look at the actual gender difference?
Yeah, yeah.
Because it's like, you know, and everybody's like, this is what the gender is.
It's very complicated, very complicated. We're 100% interested in it because I do think that's going to reveal why decades of research hasn't been published.
Yes.
The young lady here from the gerontology research, well, not so young lady, but her, she was talking about how people, researchers didn't use women in studies in pre-2005. Didn't. So everything that we think we know, only half the population. Throw in that population and then parse out all the nuances within the genders, you're in a completely different realm. Yeah. What I will say is that it does no.